Hi children, good morning everyone. Today we can study the new topic post-fertilization events in plants. We already completed the fertilization, that is double fertilization in plants. Now what will happen to this zygote as well as the primary endosperm nucleus and other things we can study in this topic. Okay. So post-fertilization events, it includes development of endosperm and embryo, maturation of the ovules into seeds and ovary into fruit. All these events comes under the category post-fertilization events. First, we can move to the endosperm development. We already learned during double fertilization, the central cell is there. That is primary endosperm cell. This cell undergoes continuous division. As a result, the primary endosperm cell becomes primary endosperm tissue and it is filled with nutrients or the food reserves. Because you know, this endosperm nourishes the developing embryo. And later, the primary endosperm nucleus, this also undergoes repeated division. But the one important point here is, it is free nuclear. The first divisions are free nuclear. Means, only the nucleus undergoes division. There is no cell wall formation. Okay. As a result, a free nuclear endosperm stage is formed. Have you seen the free nuclear endosperm? Yes. It is nothing but the in tender coconut water, you are familiar with that. Tender coconut water is nothing but the free nuclear endosperm. Okay. After that, cell wall formation occur and it becomes cellular endosperm. It becomes cellular endosperm. The white kernel inside the coconut, that is the cellular endosperm. Okay. And here, the endosperm development begins earlier than embryo development. Why? No, endosperm is filled with nutrients. So, first the endosperm development completes. After that only the embryo development begins. Because the embryo gets the nourishment from the endosperm. Now we can move to the embryo. Embryo development. So, as a result of double fertilization, a zygote is formed. A diploid zygote is formed. This zygote undergoes division. First, it becomes, this is the zygote, and after the first division, it becomes double. Now, two cells you can see here. Now, later it becomes another stage called globular. This is the globular stage of embryo development. Then it becomes heart-shaped embryo. Okay, it is known as the heart-shaped embryo. After that, it becomes the mature embryo. Mature embryo. In this way, the development proceeds. First, zygote is there, it is single cell. Later, it undergoes successive division and there are many stages, sub-stages are there. Globular embryo, heart-shaped embryo and the mature embryo. Now, we can study the structure of dicot and monocot embryo in detail. You already learned these things in the class 11 also. In the case of, it is the structure of a dicot embryo. Here you can see two cotyledons. Okay, two cotyledons and one embryonal axis you can see. In dicot or dicotyledonous plants, you can see two cotyledons and one embryonal axis. Two cotyledons and one embryonal axis you can see here. These are the two cotyledons. And this is the embryonal axis. Above the embryonal axis, this part is known as the epicotyle, which terminates in plumule. This structure is known as the plumule. Later it develops into the shoot. So this embryonal axis, the upper part is known as the epicotyle, which terminates in plumule. But the lower part, that is known as the hypocotyle, which terminates in a structure called the radicle. This radicle develops into root, and it is surrounded by, there is a cap-like structure called the root cap also you can see. So these are the structures of dicot embryo, two cotyledons and embryonal axis which contain plumule that develops into shoot and the radicle that develops into root. Okay, it is covered by a root cap. Now we can move to the monocot embryo. Here only one cotyledon is present. In Grass family, the cotyledon is known as scutellum. The cotyledon, a single cotyledon is there which is known as scutellum. And here also you can see the embryonal axis. The upper part, like the dicot, the upper part is epicotyle. There it is the shoot apex is there, which is covered by certain structures called the coleoptile. And at the same time, the last part, the end, here you can see the radicle which is covered by certain undifferentiated layer called the coleorhiza. That is an important part. So here the cotyledon is known as scutellum and the shoot apex is covered by coleoptile and the root apex is surrounded by coleorhiza. Okay. Now the next part is seed. How the seed is formed? From which part the seed develops? The fertilized ovule that changes into seeds. Okay. So here, in some seeds, if we examine in some seeds, for example, pea, beans, groundnut, etc., the endosperm is completely consumed by the embryo before seed maturation. Okay. And such seeds are known as non-albuminous seeds. Why it is called non-albuminous? Because there is no endosperm. It is completely consumed by the embryo before maturation of the seeds. Whereas in the case of coconut, castor, etc., some amount of endosperm is still there in the matured seed. They are known as albuminous seeds. It is very important. Non-albuminous means there is no endosperm in the mature seeds. But albuminous means some amount of endosperm is there. And mucellus also you know, the undifferentiated cells in the ovum. So this in some cases like pepper, beet, etc., this nucellus also persists there. That is known as perisperm. The persistent nucellus is known as perisperm. Okay. And the seeds are very important, you know, because in our agriculture is entirely depend on the seeds. And these seeds has another important property. They can dehydrate. They lose their water, reduce the amount of water, and the whole metabolic activity reduces, and it enters in a state of inactivity called dormancy. That stage of inactivity of the seed is known as dormancy. So in this dormancy stage it remains as such. And whenever it gets favorable condition, the temperature and everything, oxygen availability, everything becomes favorable. It starts to germinate. Okay. This property can be utilized in our agriculture because we can store the seeds for a longer period, for the next year also. We can use it as our food and we can use the same seed for growing the crops in the next year. So it is a great advantage. Okay. Now, this is the seeds. Now what will happen to the ovary after Fertilization. The ovary changes into fruits. Ovary changes into fruits. Okay. But in some cases, normal case, the ovary becomes a fruit. But in some cases, other parts of the plants also changes into fruit. For example, in the case of apple, okay, this apple, strawberry, etc. It is not the ovary that becomes the fruit. It is the thalamus part. Thalamus, you know, that thalamus develops into fruits. So this is not the true fruit. So we can call them as false fruits. So apple, strawberry, etc. These are not true fruits. They are false fruits. Why? It was a previous board question. Why apple and strawberry considered as false fruit? Why? Because they are developed from thalamus, not from the ovary. Okay. And at the same time, there are some plants. For example, banana. This here in such plants, the fruits developed without fertilization. So fruits formed without fertilization. Normally, as a result of fertilization, the ovary changes into fruit. But in some plants like banana, fruits are formed without fertilization. Such fruits are known as parthenocarpic fruits. Parthenocarpic fruits. So there are three categories: true fruits, false fruits, and parthenocarpic fruits. True fruits means these fruits developed from ovary after fertilization. False fruits means these fruits developed from other parts. For example, thalamus. And thalamus changed into fruits, and such fruits are known as false fruits. Example, apple, strawberry. And fruits formed without fertilization are known as parthenocarpic fruits. Okay. Example, banana. Now, this is the case of fruit. There is another possibility: formation of seeds without fertilization. Formation of fruits without fertilization is parthenocarpic fruits. But formation of seeds without fertilization. Such seeds are known as apomictic seeds. Or this process is known as apomixis. Is very important. Apomixis means development of seeds without fertilization. Okay, and such seeds are known as apomictic seeds. And how these apomictic seeds are formed? You know, egg cells are formed by reduction division, meiosis. Normally eggs are haploid in nature, so it is formed by reduction division. But in some cases, without reduction division, egg cells are formed. That means they are diploid. They are diploid egg cells. So these diploid egg cells directly developed into seeds. After these are diploid egg cells develop into embryo, and inside the seeds are formed. Because these seeds are formed without fertilization. So actually it is asexual. There is no sexual formation. It is asexual. But seeds are formed. Seeds are the result of sexual reproduction. So actually it is asexual reproduction. It mimics sexual reproduction. That is apomixis. And in some cases, like citrus, if you use the orange or lemon, you can see many inside the seeds. You can see many embryos. Okay. How this is formed? The nucellus surrounding the embryo sac. Sometimes it undergo repeated division and it enters inside, it protrude inside. It is the embryo sac. So here the nucellus you can see. This nucellus undergoes continuous division, repeated division, and it protrudes inside the embryo sac. And each one become an embryo. So inside a single ovule you can see many embryos. Such condition is known as polyembryony. Polyembryony. Occurrence of many embryos, that is known as poly. Poly means many, polyembryony. Example, citrus. So this apomixis and polyembryony both are very important in your board exams. And the last part is advantages of using this apomictic seeds. Normally, farmers are using hybrid seeds nowadays because in hybrid, cross hybridization is there. As a result, the different characters are combined. Okay. But here the problem is, if a farmer is using hybrid seeds, the problem is every year they have to produce because the same seeds they cannot use. If they collect the seeds from one progeny and use it for the next generation, they will not get the correct result because of segregation of character. That you will study in the genetics chapter. There is a process called segregation. The characters may segregate, separate. So it will not produce the correct character. And it is producing the hybrid seeds every year, buying it. It is so costly, it is so expensive. A normal farmer cannot afford it. So the problem can be solved by using apomictic seeds because it is produced without fertilization. So there is no segregation of characters. The same seed we can use generation after generation. It is very cheap and there is no need to buy every year. So that is the important advantage of using apomictic seeds. Okay. So all these changes, development of embryo and endosperm, development of seeds, fruits, all these comes under the post-fertilization events. So I hope all of you understood these things. So thank you all.